Please, thank you. So thank you, Dr. Ishihama and Dr. Amy. And yeah, now we are almost ready for the talk today. So I think all of you know the modern analytical technique called mass spectrometry, right? And especially if you use mass spec with many proteins simultaneously, it's called proteomics. So today you will hear from the expert of the proteomics. And the expert who is with us today is Dr. Yasushi Ishihama. So Konnichiwa Sensei, welcome to Bangkok, Thailand. He is a professor from Graduate School of Pharmaceutical Sciences, Kyoto University in Kyoto, Japan. And it's also the coincidence that we have his former student with us. And you guess who? Dr. Prel, Dr. Pasravin is his former student when she did her PhD in Japan. So today, Dr. Prel will introduce Dr. Ishihama, her former professor. Welcome, Dr. Prel.
Thank you very much, Ajahn Kong. So I would like to introduce his background. Professor Yasushi Ishihama graduated from Kyoto University. In his bachelor degree, he did organic chemistry, and he did pharmaceutical sciences in 1998. Then he worked in a very big pharma company in Japan, namely Eisai. And he also had a postdoctoral experience in University of Southern Denmark at Denmark. After that, he became professor, and in 2010 until now, he became a professor at Graduate School of Pharmaceutical Science, Kyoto University. Not only the roles in the university, now he served many important roles in the Proteomic Society. He is the former president of Human Proteome Organization of Japan, and now he serves as vice dean of Asia Oceania Human Proteome, or AOHUPO. Also, now he is the president of the Mass Spectrometry Society of Japan, or MSSJ. So in the lab, he focused on proteome science consisting of mass spectrometry, nanoseparation science, computational science, and cell biology to develop the methodologies for comprehensive analysis of cells and further application in pharmaceutical science. So today's topic is overcoming challenges for experimental and computational analysis of human proteoforms by shotgun proteomics. Will you give a loud applause to Professor Ishihama?
Thank you for a nice introduction, and also thank you very much for inviting me here. It's my first time to visit Thailand, so I'm really glad to be here. Yeah, so so today I'm going to talk about the proteomics from the view of the more analytical and computational aspects. And because the proteome is a really huge field, and it's actually the most, I believe that it's the most important molecule, biomolecules in our body to explain how functional we are. So just a self-introduction at first. This is my Chinese letter. So this is my family name, and this is my first name. Actually, it means that it's Thailand. So you may recognize that. Anyway, I was born in 1967, and I have one wife, two daughters. And yes, and actually I graduated from the Faculty of Engineering got a master degree from this department. But I moved to pharmaceutical science. Then I was in the industry, pharma company for 14 years. Then I switched back to academia, then now it's 18 years. So this kind of career is very, very unique in Japanese like academia field. So, and also my target was small molecules for a long time ago, but now our focus is protein. And I got PhD based on the capillary electrophoresis at that time. But now I'm focusing on more nano LC-MS to analyze the proteomes. And you can see my first daughter born in 1999. At that time, I was in the field of capillary electrophoresis. Then the electrophoretic mobility always use this mu. So that's why my first daughter name is mu. And the second daughter, at that time, I already switched to the proteomics using LC-MS, nano LC-MS. That's why second daughter name is Nano. So I didn't say anything about this to the daughters, but I just secretly, always smiling about this name in me. And these two daughters actually refused to use the current photos because they are very naive age. So I just used the photo 10 years ago. All right, so in 1999, at that time I was still in the small molecule field, working in the pharma company. So that's why I did some work about the FDA application to get the approval from FDA. So this is one of the drugs I helped to launch, and this is for PPI. It's a proton pump inhibitor, and this is really nice. Actually, work with kind of very to get the approval. It's very hard work. So we have to be very theoretical, very we need tons of the experimental data to make them confident for this drug. So this is a very unique experience for me. Then these are also the small molecule I analyzed at that time. And this is the left inside is natural product and having the anti-cancer activity from the halcyon ring. It's the creature at the seaside. And it is already also launched and named as Green. And it's completely synthesized by using the medicinal chemistry. And the right inside is antisense oligonucleotide, also having the antivirus activity. And these are very difficult to analyze at that time. But this is actually fun to analyze it. Then in 2000, I think big wave occurred. You may notice that maybe most of the students is before your birth. But so at the end of the previous century, we had a very big project called Human Genome Project. So to sequence everything about our genome. And after that, someone expect that the next generation will be post-genomic world. So then I just led one literature, which is just in front the editorial corner at the Analytical Chemistry Journal of Analytical Chemistry. And the one professor in Denmark, Matthias Mann, said that now it's the time for analytical scientists to go into the biology because the target molecule, the number of target molecules is huge. And definitely we need to develop something new to solve the next generation problem. So I just decided to go into his lab during working in the pharma company. So I spent 2 years in one lab in Odense in Denmark, and they are very, very diverse. It's very like multi-international circumstance, and also with the very diverse background. Not only the biologists, some of the guys have the very strong analytical background, and also some has the very strong bioinformatics background. So these are very nice combination to achieve something new. So I liked a lot about this circumstance. So I just summarized the, what the new aspect in the life science in this century. So just before this century, we completed the human genome sequencing as a draft format. But then it's announced at the February 2001 from the President of the United States and also the Prime Minister from the UK. So because this is a huge impact news for everybody in the world. Then after 2 years, complete human genome was opened. Then the new era starts. So what happened? So then tremendous amounts of the new finding occur just for like 15 years, like human iPS cells, like genome editing, and next-gen sequencers, and single-cell spatial transcriptomics technology, and also cryo-EM to solve the protein structure. So these kind of things actually open the new era, especially for drug development, drug discovery and development. So this is our current situation in our graduate school. So we, after like human genome completion, we actually created a new division called bioinformatics for drug discovery, because the genome itself has like the collection of the huge amount of the information about our functions. At that time, we believe that. And actually, some of the information is really nice to find the new drug target. It's like orphan GPCR receptors, for example. And this works very nicely. But then, something happened for next 20 years. Then we decided to open another new division called Medicinal Frontier Sciences. And actually, I'm now the head of this division. And because there's one big issue in the breakthrough in 2017 for the AI. I will explain later. So my task is to do omics measurement beyond genomics. This is my current task. So this is the this is the flyers for our symposium to announce that we will have the new division. And as I said, my task is now comprehensive biomolecular measurements by proteomics, mass spectrometer, beyond the genomics. So, as I said, 2001, it's just after the draft sequence of the human genome. People expected a lot, especially in the medicine field, to have the new treatment or to understand the very difficult diseases, also to develop the new drugs. So, but most of them actually failed. And some of them actually succeeded, but most of the cases we failed. Then 2010, Nature and Science or other scientific journal actually summarized what happened for these 10 years. And their conclusion is life is too complicated. So genome information itself is not sufficient enough to understand our human life. For example, you can see this caterpillar, and this is a butterfly. they have the exactly same genome. So if you just look at the genome, you cannot discriminate whether this is caterpillar or this is butterfly. So genomic information is of course very important, but only genome is not enough to understand the phenotype of our life. So this is just to summarize the circumstance of the genome. So if genome is something like this, the one circle, because this is very static, very stable through our whole life. Right? It never changed. But by time, age, tissues, organs, person, diseases, every aspect, proteome change. And proteome is hugely diverse compared to genome. It's always dynamic. And not only the protein expression or protein abundance, but also we have to think about other factors like proteome expression, proteoforms. So you may know that proteoform. So it's a kind of like isoform, but not only the isoform, it contains a lot of like variation in protein from the single gene. Of course, we have to think about the post-translational modification. This is after translation, protein should be modified with something. And this modification is also, in many cases, is reversible and dynamic, like phosphorylation. So, like 10 years ago, people believed that only 30% of the human proteome could be phosphorylated. But now, we have the experimental evidence that more than 98% of the gene product should be phosphorylated. So, it's more diverse than expected. And for interactome, so protein itself cannot work as a functional molecule. Protein always needs some partners. Sometimes it's like 1-to-1 relationship. Sometimes it's like 1-to-4 relationship. Sometimes it's protein-protein interaction. Sometimes it's like protein-DNA interaction. Sometimes it's protein-metabolite interaction. But anyway, it needs some interactors to be functional. Sometimes it's like accelerated. Sometimes it's like more like suppressed. And these are very, very functional and very dynamic and well controlled. And also protein, after like synthesis, it could be like moving to some subcellular portion, like within the mitochondrial and within the nuclear portion, maybe outside the cell, within the plasma membrane. So the function of the molecule is also depending on the position. So, and sometimes like phosphorylation is a trigger to move one place to another. So, and also some of the partner will affect the localization of the protein. So everything is well connected to be functional. And these information are not coded in genome. So that's why we have to analyze the proteins. And another aspect of the protein, so some of the people believe that transcriptomics is enough to monitor the protein abundance in the cell. Actually, it's not true. So, as you can see here, this is the correlation between transcript and the translated, we call translates. This is ribosec. You may know the ribosome profiling approach. It's ribosome always contain the mRNA. So this is like during synthesis of the polypeptide, mRNA like bited by the ribosome. So we just isolate the ribosome to sequence the mRNA within the ribosome. So it means that it's during translation. So it's the evidence that this mRNA portion like during translated. But actually we don't have any evidence that this is really translated or not. Because now we know that ribosome just stopped during synthesis, during translation. So it means that only the ribosec data is not enough. Also to monitor the synthesis process of the protein. So the center one is transcript versus protein. So this is the protein abundance. And the correlation is actually a little bit worse compared to transcript and translated products. The last one is the ribosec versus protein. As I said, the translated product is not related to the proteome cell. There are two possible causes. One is maybe ribosome just have the mRNA but not move anymore. So it's not translated. Another cause is actually protein decomposition, degradation. So protein amount within the cell should be controlled by the synthesis and degradation. It's the balance of these two factors. So that's why we have to also measure the decomposition or degradation of the protein to monitor the exact amount of the protein within the cell. So these three correlations always emphasize the importance of the experimental evidence of the proteome. So I use this slide for more than 20 years now, just to show that the protein is the most important molecule, as I said. But this is highly biased. Maybe you can believe or you cannot believe. It's up to you. But I think this is true because protein is the molecule to drive the cell. So that's why if something happens for protein, then our health condition become worse, or become better, or become worse. Anyway, protein is the like central molecule to control our health. And also, that's why the target molecule of the drug is actually the protein. Maybe not 100%, but maybe more than 98% drug target should be protein. And not only the mammalian cell, but also like plants and bacteria, the mechanism is the same. We also have to think about the post-translational modification of the protein for functional understanding of the like bacteria or plants. So nowadays proteome technology become better and better. Maybe I don't have to say this one, but like 10 years ago, always biologists, especially biologists, claim that proteomics is not enough. So that's why they want to use transcript mix, because the coverage is like almost complete, and they can do a like single cell analysis. They can do a like spatial analysis as well. But nowadays, proteome technology become better and better. Now, I think the single cell is also possible, and some of the spatial proteomics already appear. Maybe it will develop more within maybe next 3 years. So, one example why protein is really important for drug development, drug discovery and development. This is one of the reasons I actually involved this project when I was in the pharma company, and we found this very interesting natural product having the very strong anti-cancer effect. But we don't know how it works. And we need to identify the target molecule And using the genome information, it's impossible. Using the transcriptome information, we cannot identify anything. So that's why we start to do some kind of protein level experiment. We did a lot of things, but finally we decided to use the radioactive compound to estimate the subcellular localization first. Then we found that the target subcellular localization is nuclear fraction. Then we did a lot of things to identify the target protein complex. So using the immunoprecipitation and using a lot of stuff anyway. And mainly LC-MS is always was very powerful to identify the target molecules. So I will skip this, the main part, but the conclusion is this is the target molecule, sub-130, but very interestingly, if we lyse the cell, we cannot catch this molecule by using the compound. So compound sub-130 affinity need this kind of complex within the cell, only within the cell. So this is very naive because the complex is very dynamic and the molecule combined to sub-130 only within the cell. Once we lyse the cell, the complex becomes a little bit damaged. Even under the very mild condition, the compound cannot fish out the complex cell. So it needs that it's well controlled. So we need more technology, but definitely all of the technology should be based on the proteome level. Otherwise, we cannot do anything. Okay, so this is actually my first paper for a long time ago when I was a master course student. So as I said, I did the capillary electrophoresis at that time, and I did something called Edelman sequencing. The older people noticed that the Edelman sequencing is the way to sequence the protein. It took a long time, and it needed a huge amount of the sample, and so the throughput is so low. Normally it's like 1 week, 2 weeks to sequence one protein. Then, almost the same time, mass spec appeared with for the peptide sequencing. So tandem mass spectrometry appeared at that time with the very precise capillary LC separation. So the researchers in the University of Virginia identified the MHC class 1 peptide without any information. So it's de novo sequencing for the peptide level. So in principle, we use this technology even right now. Even now, we still use this tandem mass spec based peptide sequencing. So I will explain how we can identify the peptide sequence using the tandem mass spectrometry. So it's a kind of like analytical chemistry lecture, but I hope that you may enjoy. So this one sample means amino acid, and if it's connected like 6 sample, it's like 6 amino acid residue peptides. So after protein digestion by trypsin, you can get this kind of small fragment, the peptide, and we just separate this peptide by chromatography, but then do electron spray ionization to go into the tandem mass spec. So tandem mass spec, there are 2 rooms. The first room to filter out to discard the untarget molecule. Just keep the target molecule, target peptide. Then in the second rule, maybe some kind of like argon or other inactive gas just attack to the molecule. Then the molecule become like decomposed, but not completely. So this is a kind of like, how to say, if you have like 7 amino acid residue here, then after collision, you can get this kind of ladder sequence. We call ladder sequence. It's just one amino acid decomposed one by one. Then if you can measure these like 5 molecules by mass spec, then you can immediately see this one, which amino acid is this one, which amino acid is this one, based on the mass of the amino acid. So you may remember that how many amino acids in our body has? How many? Like natural amino acid. We have 20, right? So among 20 amino acids, how many amino acids has the exactly same mass? So of course structure is different, but how about the mass? Only one pair, leucine and isoleucine. These has the exactly same mass, but others we can discriminate just by looking at the mass. So that's the basis of the mass spec-based proteomics. So this is just like without collision. This is a mixture. Just go into the tandem mass spec. You can see a lot of peaks. So we can just select one of the target peptide, which is like 829.013 mass peptides. Then just isolate using the MS1 part. Then discard everything. And react with the argon gas. Then it becomes chloride. And you can see like very distinct peak. The biggest peak is here. And the difference between first one and second one is corresponding to valine. And that's the same for the second amino acid. And third one is glutamic acid. And fourth one is also glutamic acid. So these are like the partial information about the amino acid sequence of the target peptides. Now, we should use other information, like we use the trypsin to create the protein. Then the C-terminal of this target peptide should be lysine or arginine. And also for N-terminal, next to N-terminal, this amino acid should be also lysine or arginine, right? Then, as we know that this is BVEE, but we already know the entire mass here, and also entire mass here. So, and we are also measure the 899.013, so we know the entire mass. Then these kind of like information just combine, then search against the in silico database, human protein database. In silico digested at the lysine arginine to produce the in silico tryptic peptide. Then just match between experimental one and in silico one. If the length of the peptide is more than 7, you can uniquely identify the human proteins. Only 7 amino acid residue will be enough to be unique in human proteome. So that's also the big advantage to use the trypsin. So these are the like basic strategy to use the LC-MS-MS based approach. So as I said, this is my first paper published in 1991. At that time, it's Edelman sequence technology. Now, currently, the speed and sensitivity is 1 million times more. So it's huge improvement. So within 30 years. So before, so at that time I'm here. So let's summarize the approach for LC-MS. If you have the protein mixture here, digested by some protease, maybe most of the cases trypsin. Then this tryptic peptide mixture are injected to the LC. Most of the cases nano-LC. Then separated in time. Then shoot into the tandem mass spectrometry by electrospray ionization. And you can get this kind of ladder sequence. And this partial information can be used to database searching to identify the protein. Okay, so let's see the situation of the genomics. So next generation sequencer is really powerful, as you may know. And because they just discard the electrophoresis process, now they don't need to separate the molecule in time. So that's why it's very fast, and also it's very easy to parallelize. And it's a kind of a game changer. Everything changed by. So this is the a little bit old slide that you can see here. This is the cost of the amino acid sequencing or DNA sequencing. And this is the year. The blue one is actually the genomics. And you can see the big drop here. And this is because of the NGS. So as I said, NGS is really real game changer. Not only the research field, but also entire society. So others are LC-MS-based proteomics. So it's increasing. It's decreasing very constantly, but definitely we need something else to get the same drop. Then maybe instrumental breakthrough or computational breakthrough or completely different technology like nanopore sequence. And we don't know right now, but I still believe that the mass spec-based one is more versatile because it's mass-based. So every molecule should have the different mass. Of course, some of the cases it's not true, but and this should be very powerful. And this is showing the progress of the proteomics based on the LC-MS. And from 2001 to 2006, by using the yeast proteome. And these are theoretical distribution of the proteome abundance. And x-axis is the molecules per cell. And y-axis is the frequency. So these are highly abundant proteins. These are very low abundant proteins. And using the lower model, of course, the coverage of the highly abundant protein is higher compared to lower part. And it becomes higher and higher. Then, in 2008, actually for yeast proteome, it's completed by using LC-MS. But it takes 41 days only for single sample. 41 days, it's too much. But then technology also become improved a lot, and I don't know now, it's 2014, it become just 1 hour. So the technology always go ahead. So I didn't show the most current performance of the mass spec, but now it's 10,000 human proteins within 1 hour is possible. So it's huge different from the past 10 years. But then what we need in the future is like these three aspects. Like depth of analysis or like coverage of the analysis. And also we need the sensitivity to measure the single cell proteome or subcellular proteome at the single cell level. And also we need throughput. Of course, like clinical sample, we have tons of sample, right? So currently, on average, we always use like 2-hour analysis time for one single sample. Then the throughput is like only 12 samples per day. If you have like 1,000 sample in the biobank or cohort or like disease-specific data bank, it's impossible to measure everything. So definitely we need the throughput as well. So the instrument we are currently using is the called not the tandem mass spec, but the ion mobility device with tandem mass spec. So this ion mobility device called TIMS is trapped ion mobility spectroscopy. And so normally we use the LC in front of the mass spec, and the LC separation is based on the second. And TIMS can separate the ion within the 100 milliseconds. And the quadrupole, like fast MS1 separation, is based on like 10 milliseconds. And the final part is top, is time of flight mass analyzer. It needs less than 1 millisecond. So this is a very nice combination. So we got this instrument from Bruka. This is actually the first instrument in Japan. It's like 6 years ago. This is actually the German product. So German guy, I think the average height is a little bit, not limited, but much higher than normal Japanese. Especially in Kyoto, we have very strict rule for the height of the building, because we want to keep the view from the city center to the mountain. We should keep the view. So that's why all of the buildings should be less than 5 floors. So then each floor, the height is not enough to get the German product. So we have to remove the roof. So at first we considered maybe we can angle the instrument, but then the broker guy, no, never. Then we decided to remove the roof. Anyway, so this is like a chromatographic part. For chromatography, nano-LC, we also create our own column by ourselves. It's not the particle based, it's not the particle packed. It's a monolithic structure. It has a lot of advantage over the normal conventional particle pack. But so we can get this kind of separation very nicely, but it always take a very long time because we use a very long column, like 2 meter column. Then we can get much higher number for detection. But it's very, very difficult to get identification based on the MSMS spectra, because we use a very long time for separation, and for each peak, it's separated with each other, but for each peak, actually, it's already diluted. So we have to concentrate the peak, even sacrificing the separation efficiency. So that's what we need. So maybe I will skip this part. So this is the current situation of our field, and there are 3 factors, as I said, like throughput, sensitivity, and depth of analysis. So the x-axis is a throughput. y-axis is the depth of analysis. It's like identification number. Then the circle size is the sensitivity, injected amount. So we just summarize the current literature. As you can see here, the ideal part is right upper corner. Higher throughput, higher depth of analysis, and also with the smaller circle. But clearly there's some limiting line here. So we should go into this region. So that's our goal. So before that, we analyze the effect of the flow rate. So this is very very technical aspect of for electrospray. So electrospray need lower flow rate to ionize everything. If you have the higher flow rate, it's impossible to ionize everything. So we need to slow down the flow rate. And the flow rate effect is so huge. So we should keep the flow rate less than 10 microliter per minute. So we also create the very sharp gradient to make the peaks sharper. And in this case, we use just 1 minute gradient. So before that, we always use 8-hour gradient, or sometimes 48-hour gradient. But now we decided to minimize the gradient time to get sharper peak. So we just accept the 1 minute gradient with less than 10 microliter per minute. So this is like the standard condition using the particle packed clamp, 500 nanoliter per minute, and a little bit more than 1 hour gradient. Using the 3 synthetic peptide, the sensitivity is something like this. When we use the monolithic clamp I introduced with the exactly same condition, but use the higher flow rate, as expected, the sensitivity drops. But the retention time becomes a little bit shorter because of the higher flow rate. When we use the very sharp gradient like less than 1 minute gradient, now the gradient pump cannot produce the reproducible profile of the gradient. So that's why the peak split. But if we use the 10 microliter per minute, we can successfully having the 0.8 minute gradient profile. Then we can get very nice peak. It's almost 1 minute within 1 minute. So this is the overlay between this one and this one. And now you can see the gray one is in minutes, and the red one is in second. So almost the same sensitivity obtained, but a much higher throughput we can do. And this is the entire system now we are developing. So this is very technical one. I don't know if you have enough LC knowledge, then you can understand why the gradient mixer is here, and it's better as close as possible to the injection valve, and injection loop is 1 microliter, and it's directly connected to the analytical clamp, which is the silicon monolithic clamp. Then the gradient delay is less than 15 seconds. And the clamp pressure is much lower because because of this monolithic structure. And the clamp length is 150 millimeter. So 100 micrometer ID, 150 millimeter. So it's a kind of nano LC clamp. But we can operate under the capillary LC condition, which is more than around 10 microliter per minute. Then we can minimize the clamp washing time and clamp equilibration time. So this is what we are actually doing. So these 3 instruments are independently operated, and this is the old sampler, and there's a syringe inside. You can see here something moving, and there's the injection valve here, and injection valve here. So now it's started, and the gradient also started like this, and until 48 seconds, the gradient up to 40% of the acetonitrate. And during this, the auto sampler is still busy to wash the syringe and also ready to suck up the next sample. You can see maybe here, now the sample tray open, then the syringe can aspirate the sample into the syringe. Then maybe I will stop that after 1.4 minutes, the next analysis start. So it means that the throughput, LC cycle time is 1.4 minutes. It's 84 seconds. So one sample just needs 84 seconds. It means that we can analyze 100 samples per day. So this is much higher throughput compared to the existing technology. So maybe I will skip this part, but I will show this. We are using the data independent analysis mode for data acquisition. So that's why it's very strong to accept the co-eluting peaks. So this is also another benefit why we can use this very short gradient. And the identification number from the HeLa cell lysate, when we inject 100 nanogram into this system, we can identify more than 3,000 proteins. So 1.4 minutes analysis can produce the 3,000 protein ID. So this is also very nice, and even less amount of the injected sample is like 10 nanogram. Even 10 nanogram, we can identify more than 2,000 proteins with 1.4 minutes. So things are really different from like 10 years ago. Now it's, I just showed the same slide, but with our achievement. Now our achievement is here. So as we hope, we got this area. So now we are still working to increase the throughput more. So we also analyze analyze the stability of the system. So actually, when we submit this manuscript, then the reviewer wanted us to do like 1 week continuous analysis. So then like 5,000 samples per week continuously. So it's really big challenges because you may know that the mass spec itself is not so stable. And also the column itself is very easy to be clogged during analysis. And there are a lot of things we have to take care for 1 week without stopping the system. So, but we did very nicely. As you can see here, we used the 2 different acquisition mode with 2 different throughput and using the 3 different sample plate containing 96 samples per plate. So we successfully analyzed. Very unfortunately, the system stopped just before the end of the analysis. So we are so disappointed, but I think we just report as it is because this is the realistic result. So skip this. So another aspect is the carryover. So from the analytical chemistry point of view, the carryover is a really important point to measure the quantitative analysis. So as you can see here, this is the HeLa sample, and this is the blank sample. You cannot see any big peaks here. So we have a lot of tricks to get rid of the carryover peaks. So now it works very well. So we don't have to worry about any carryover, even for like 1,000 different samples. Okay, so Then this is one of the applications we are trying. So for transcriptomics, of course, the spatial transcriptomics is very powerful. So we also want to do the same thing. But the problem is the sampling. So in this case, we just use the mouse brain slice and use the laser microdissection. And the pixel size is not so small. It's 400 micrometer square. So, but still, we can get the 96 sections and analyze by this 1.4 minutes gradient, 1.4 minutes analysis, and it took only 2 hours to analyze everything. So this is the result. So we use just half of the part of the mouse brain, and so this is 8 times 12 sections, and these are the number of peptide, protein groups, and so on. But what we are interested in is to see some cell-specific markers. And this is the oligodendrocyte markers, and this is the nearing binding proteins. And so these peptides are from this protein, and these peptides generate exactly same distribution. So also with the different proteins, but also the marker of the oligodendrocyte showed exactly same distribution pattern. So it means that we can successfully monitor the localization of the protein based on the cell distribution, cell type distribution. And this is also true for cortex biomarkers. So, of course, we have to increase the spatial resolution in future, but in general, the strategy works. So, another application is, of course, the clinical. So, now we can analyze 1,000 samples per day. So, we are now planning to do very large cohort analysis, maybe 20,000 samples, and this could be done within 1 month. But we also want to increase the throughput more. So the current plan is 10,000 samples per week, and now the system itself works. So we are planning. But anyway, this is preliminary experiment to see how it works for plasma proteome. And you may know that the plasma proteome is one of the most difficult samples, because you may know the top 20 highly abundant plasma proteins occupied everything. More than 98% of the protein are from these top 20 abundant proteins. So that's why it's very, very, very difficult to identify the low abundant protein except these top 20 proteins. And this killed this technology many times for a long time. Since 2000, many people tried, but they failed. So this time we just look at the peptidome, not the proteome. So just use the soluble fraction after the acetonitrile precipitation of the plasma. So this is just a preliminary one, and we found that most of the ultra high abundant proteins are depleted by using this technology. But also we can find very interesting peptide, bioactive peptides. And so this is the COVID-19 samples. The peptide content from the patient is much more than the healthy volunteers. It means that maybe protease will be more active in the patient because there are a lot of inflammation. So, but the good thing is we already have some candidate as a biomarker, and we cannot show the real peptide name, but we found one of the C-terminal fragment of the target protein increased a lot during the COVID-19 infection. So we are now very happy to see this, and we are also continuing this project right now. I will switch gear a little bit to more computational one. So as I said, we need two types of the innovation for the improvement of LC-MS-based proteome sequencer. So the second one is the computational breakthrough. So you may know that this is the cryo-EM, and it's awarded by the Nobel Prize in 2017. And during this ceremony, actually these documents are open, and I read this with very impressive feeling that this is the old days resolution by using cryo-EM. And this part is the current resolution. And for instrument-wise, nothing changed. This is because of the statistics. So because now we can use big data to estimate something based on the statistics. So that's why this high resolution is realized. So I saw that the same or similar approach can be used for our LC-MS. So that's the trigger to start this project. And this is just summarize how to infer the cause from the result. So this is the result. And the original picture will be in the left. And I use some tools to create this beautiful board. And this is the course, and we use a special tool to make this space beautiful. And this is impossible if you have this one example. But let's imagine if we have a million example, 1 million example, then we can estimate what happened during this treatment, right? So there's some equation, maybe there are some equation to convert from the one picture to the converted one. So maybe variable number is not 1 million. Maybe variable number will be 100. Then if you have 10,000 example, then it's very easy to estimate what kind of variable they used. So this is the power of the large data set, big data, to estimate what happened. Yeah, so the big data is the key. Also, the statistics is the key. So, and this is what we are doing for proteomics. One sheet contains the amino acid sequence for single protein. If you have 10,000 proteins, the number of sheets is 10,000. So we can use shredder to make the piece into the peptide. And also we can use some more stronger shredder to make the peptide fragment. After that, we can estimate what is the original sheet just by combining small pieces into this piece. Because the cutting phase, it needs to contain some information. Then based on this, shape and the content, you can create this sheet. But the success rate is not 100%. We always get around 30%. So this is our reality. So we can use another technique with the help of statistics. So we use the hierarchical Bayesian models, which are already established. I'm not so familiar with this, but I just got this information from our collaborator, and I explained what we are doing. Then he recommended us to use this hierarchical Bayesian model. So I think now you are already understanding this part. It's LCMS spectrogram. So we call spectrogram, it's a spectrum depending on the time. So the x-axis is m over z, is a mass divided by charge, and the y-axis is the time. So this is like standard LCMS MS spectrogram. And we just pick up one of the peak to do MS. So we have another phase of the spectrogram, but anyway, this is a kind of like standard profile from LCMS. If you cut one particular time, then you can see MS1 spectrum, and also using one of the peak in MS1 spectrum as a precursor ion, you can get the product ion spectrum, MS2 spectrum. So each peak has this kind of like isotope cluster and also charge distribution as well. So what we are doing now, and not the statistic one, like conventional way, once we got this spectrum, we just set the threshold to make the spectrum very simple and convert into the just number, the table. This table could be used to do a database search against the in silico protein database. Then we got the estimated peptide sequence, but the complexity is not of the peptide protein relationship is not so simple. So one peptide shared by two proteins, this is very likely, or shared by three proteins. But these three proteins, it's not the same sequence, partially same, but the entire sequence is different. It means that two proteins has their own unique sequence and also common sequence as well. And then there are also a huge number of cases, then it's very difficult to estimate the original proteins based on the peptide. So this is the problem we call protein inference problem. And we have to assume something, otherwise we cannot determine. So that's why, as you may notice, that I did not say protein identification. I always say that protein group identification. So it means that we cannot specify which protein within protein group. We can identify protein groups, but we cannot identify each protein. So this is a protein inference problem. But, so because we firstly identify peptide, then do protein identification. So maybe using some nice statistics, we can use both. So the collaborator we work with is Professor Kazuyoshi Yoshi. He was here for 2-3 years ago. Last year, I have no. Last year, you may then remember that he also gave the presentation about this. But his measure is not the proteomics. His measure is more interesting one. It's like sound separation. So if within the same meeting room, 5 different people say different things, just because everything after recording, his technology can separate based on the person, 5 persons. So this technology can be also used for music. If they can record, he can record the orchestra, for example. Then different instrument used. Then record everything, and using his technology, separate into each instrument to make the original score. So, and it means that you can delete the, for example, vocal if you don't like her or him. Then replace the new voice. That is also possible, and that's what he did in his homepage. So we discussed a lot how to apply his technology to LC-MS. It looks completely different, but what we noticed actually looks very similar in terms of the data structure. So as you can see here, music data and proteome data looks very similar. It's always time scale. It's always, sometimes it's a frequency for music data, but the proteome data is M over Z. And there's a huge number of rules, very restricted rule. As I said, amino acid number, the kinds of amino acid is only 20. It's not 25, it's 20. And also, what happened? Zoom. Connecting. Okay. Can you hear me? Zoom people? Oh, yeah, but if I use it. Yeah. It works, right? Okay. Can you hear me? All right. So yeah, so XYZ axis is very similar between two data sets, but also tone number, amino acid number, and also code. Right? It's like do re mi fa sol la si do and re mi fa sol la si do re, and so these kind of things are also well controlled for music data, but also it's controlled by the combination of the amino acid. And charge state is corresponding to the octave difference, and yeah, these kind of things are very similar with each other. So we decided to use the technology. I think that's what she did, and in her PhD. So we use this kind of matrix decomposition approach called NMF, and actually it works for our LC-MS data. Our LC-MS data we called protein matrix mass spec decomposition, mass spectrogram decomposition, and it works very well. And not only the very simple sample, but also 48 protein mixture containing 9,000 ions. We can separate the, we can decompose the signals. And finally, this approach can be used for very complex mixture like E. coli for lysate digestion, and it works also very nicely. Actually, it's better in terms of the sequence coverage compared to the conventional approach. So now we are also using this technology for more versatile data independent acquisition mode using the similar theory, and we got some preliminary result showing that it also works for very complex mixture like human protein samples. So this is one of the way to use some computational approach, but this is more like general explanation about the genome. So at first I said that the genome is not sufficient enough to estimate the function. So in terms of the functional analysis of our human life, genome is not enough. And also transcript is not enough. So we definitely need the proteome and proteoform information. But also genome has another aspect. It's evolution. So now next-gen sequencer can sequence a lot of creatures in our world. So then we can estimate how biological evolution occurs. So this information is really also well organized. And this is not possible only for proteome. I think this is the benefit to use genome information. So what we can do by using this biological evolution information based on the genome. So as I said, 2017, some big breakthrough occurred in the field of AI, deep learning. So you may know that the Google people just published one paper about the transformer model. This is based on the natural language model, language processing model, originally for automatic translation. You may notice that now the recent tool to do translation by iPhone or other tools, it become much better than 5 years ago because of this natural language processing. So this transformer model, there are some big benefit, but in principle we can also use the protein language model we call. So based on this evolutionary data of genome, so you can see the number of protein sequence is 250 million protein sequences containing 86 billion amino acids. So this is really the big data for this transformer model. Now transformer model can accept this kind of evolutionary data to learn how each amino acid or amino acid combination or amino acid sequence can transfer to the function. So and the Meta people in Meta and Facebook also publish based on this transformer model, but using this evolution amino acid sequencing data. It's already pre-trained. It's also 669 million learn expression. So it's huge data, but it can also predict the structure of the protein. Actually, this is also based, not exactly the same, but it's also based on the AlphaFold 2. So, yeah, we use this ESM1B, then input is protein sequence, then so this is already published data, not from our group, but also by other groups. So they already achieved the estimation of the secondary structure or prediction of the mutation site, and also prediction of the contact portion between two proteins. So these are possible based on this technology. So we should use to predict the ion mobility. As I said, we always use the ion mobility separation before tandem mass spec. Ion mobility is based on the collision, we call CCS. Anyway, it's the kind of like physical chemical properties of peptides. So it could be predicted by using this transformer model, but just use this existing model. It was impossible, and so that's why we changed the model to make more versatile. And finally, we got very nice correlation between experimental ion mobility and predicted ion mobility. So this is the result. And the another aspect is, as I said, it's pre-trained device. And if you want to do this very large-scale deep learning, normally you need huge resources as a computer. So it is not possible for like single lab, of course, and for entire university, still it's not possible. It is only possible for Google and Meta and Microsoft. So, but nice thing is this is already pre-trained. So we can just use this device, then combine with something new to create the nicer prediction model. So that's I think the next generation. Yes. So because my title contains proteoform, so I will show something about the proteoform. So as I said, the protein itself has some variation, and from the single gene. So this is like canonical sequence, like the typical standard sequence. But sometimes you can see, because of the RNA splicing, and the N-terminal will be different. And sometimes with the different translation initiation site, and the product will be different. And sometimes after translation, some protease just cleaved at the C-terminal site or N-terminal site, then the final product becomes different. And this endogenous proteolysis is also very famous for a lot of enzymes. So you may know that because it's pharmacy department. So there's a very famous hormone called GLP-1. So glucagon-like peptide 1 is highly related to a lot of disease, especially for diabetes. Now there's commercially available drug a lot to control the weight. So this GLP-1, so there's a GLP-1 precursor, right? Then one enzyme can cleave at the N-terminal site to be active. But also within the plasma, within the blood, another protease just cleave also some 2 amino acids at the N-terminal to be inactivated. So it's well controlled, their activity is well controlled by protease. And so the drug itself is like very mimic to this GLP-1. So that's why, but it has some, like for example, D amino acid to prevent the cleavage by protease. So that's why this drug can be used as a drug instead of the GLP-1. So anyway, this kind of like endogenous protease proteolysis is also very, very important to monitor the function of our proteome world. But at this moment, to analyze the proteoform, it's not so easy because protein itself is very difficult to ionize with the high sensitivity by mass spec. And also after ionization, to get the sequencing information by looking at the product ion spectrum, the efficiency is not so high. So that's why we have to cleave the protein to the peptide at first. So that's why we are always use this shotgun proteomics approach. But to identify the exact proteoform, I think the entire protein is needed. So also there's another problem about the protein inference problem. As I said, if we cut the protein with trypsin, we lose the information whether fragment 1 and fragment 2 are originally connected, or fragment 1 and fragment 3 are originally connected. We don't know. So the approach we are now using is in Japanese, it's a smoking pipe. So you know the smoking pipe? So the internal part is wooden, but the external part is always metal. It's very stable. It's very strong. So we call smoking pipe light for train. This is illegal approach. If you have the nice ticket only for this part and this part, you don't have to buy this part. As I said, it's illegal. But when I was young, many people use this smoking pipe light. But nowadays it's impossible because we are using this electric car. Anyway, it's illegal approach, but we just want to use this approach for protein analysis. We're just focusing on the N-terminal and C-terminal of the peptides, of the protein. So the key technology is how to isolate the protein N-terminal peptide and protein C-terminal peptide after protease digestion. So this is a kind of long-term questions in this field. 20 years, nobody can solve this problem to find a very nice way. So then people start to use a lot of series of chemical reaction to enrich the N-terminal peptide and C-terminal peptide. But by accident, we found very nice way. Just use one particular protease and just use one particular chromatography to isolate the peptides from protein terminal, N-terminal and C-terminal. So we call CHAMP. It's a chromatographic amplification of the protein terminal peptides. So we maybe I will not explain too much about this, the mechanism of the isolation, but we use the lysoarginase for digestion, then use the strong cation exchange chromatography, but it's not the HPLC based, it's just a pipette tip based. So it's disposable, and also you can use simultaneously with 96 tips simultaneously. So it's very cheap and convenient, and this is also very suitable for high throughput analysis. And very similar approach can be used for C-terminal enrichment. This case we use a V8 protease. It's sometimes called GluC, but it can also cleave the ASP-C, C-terminal side of the aspartic acid. So by using this enzyme, and also this is the metal oxide particle to capture the dicarboxylic structure at the C-terminal side of the peptide to get rid of the internal peptide. Then the rest of the peptide should be from the protein C-terminal. Yeah, so like this, and we established the method, and then we found that so combination of these three methods would give a very nice coverage of the protein terminal peptides. So now, for example, as I said, proteolysis, like endogenous proteolysis product could be captured. So using this one of the CHAMP approach, we use the I think HEC293 T cell to isolate the protein N-terminal peptide. then we found a lot of proteolysis products which are different from the canonical and terminal. It's very easy to discriminate whether this is from the canonical and terminal or newly synthesized in terminal. And we checked using the the uniprot information. Then most of them are actually based on the signal peptides, and sometimes it's transit peptide to be like mature form. But actually, we have also a lot of unknown peptides. So, yeah, as you see here, like less than half can be identified as transit peptide and signal peptide, but most of them no information at all. So then we use this signal P 6.0. So signal P is the prediction tool to predict the signal peptide portion. And this has been used for a long time, but the version 6.0 start to use protein language model. So they use the protein language model, then the accuracy becomes much higher. So I was so surprised to compare with our experimental data, how they can predict. And so this is like part of the data, as you can see here, and signal P 6.0 predict this as a signal peptide. And this study, we identify this in terminal site after cleavage. So it means that signal P accurately predict the experimental data compared to the uniprot data, which are also based on the signal P, not 6.0, maybe like older version. Oh, so this is no experimental evidence, but these are like based on the old version of the signal P, and these are all wrong. I think because of the agreement with each other, I think this will be more correct. And this is also true for transit peptide. So there are another program called Target P. So they predicted very accurately in agreement with our experimental data. And these are a little bit different from the previous version of the, which are registered in uniprot. Yeah, so so this is what I showed like at the early time. So always the protein abundance is not very correlated with the ribosome. So ribosec information. So then what I said is we have to think about the decomposition, degradation of the protein. Sorry for some Japanese. So there is the method called short-lived protein mapping by Harvard group. So they use the cyclohexamine to stop the protein synthesis, then measure how they can survive after stop the synthesis to measure the protein half-life, especially for shorter lifetime proteins. So we just combine with the technology and our N-terminal enrichment technology, then found, sorry in Japanese. So this is our result. Is yeah, we also have the technology to isolate the nascent polypeptide from ribosome. So it just synthesized, then just enriched using some special technology. Then we found that there are some like features with like acetylation on the first methionine or without acetylation, but still with methionine and acetylation without methionine, and non-acetylated, also without methionine. So these are four types of the N-terminal we can see. And if we look at the just canonical N-terminal, the profile of these four kinds of N-termini looks like this. But if we look at the non-canonical, in total, it's completely different. But also, if we combine with the nascent polypeptide, and also this is non-canonical nascent peptide, then it's also different from the canonical property. And this is also true for short-lived peptide versus other peptides, other proteins. So, the decomposition of the proteome is not so easy to estimate so far, and someone believe that there is the sequence to facilitate the decomposition degradation we call degron. But in some cases this is true, but some cases it's not true. So there's no consensus conclusion about the degron. So finally, we decided to apply protein language model to predict this endeavor is existing or not, independent of the protein, independent of the biological context. Just input should be protein sequence. That's it. Then this young student in my lab, he has some strong background about the computational approach. So we just use this ESM2 in this case. Then input is just a protein sequence, and he also added some structural information into each amino acid. This is based on the AlphaFold 2. So then estimate that, predict the protein lifetime based on the protein half-life. And we just use the published data set, one of the published data set from the mouse fibroblast data. So the prediction accuracy is more or less like 0.7. So it works. I think it's impossible to be like 0.9 because it's only the sequence based. It's not containing any biological context. So, but this estimation, the prediction model also can be applied to human model. So we also have the different set of the protein half-life data, and also the prediction accuracy is more or less 0.7 to 0.75. So we are really happy to see that this protein degradation can be also predicted by using protein language model. And then we can also get some, sorry, it's not published yet, so we can show the exact sequence, but we know some of the dipeptide or tripeptide facilitate the protein degradation very clearly. So I think that there are some integron within the sequence. But not only the sequence, actually, but also we have to think about the surrounding structure information. So that's our conclusion right now. And we are also working to collect more data which are based on the biological context. So once biological context change, then how this kind of background change. So this is the next step. But anyway, what I want to emphasize is the importance of this kind of deep learning technology. Definitely, it will help a lot. These kind of things so far it was impossible, and many people tried, but we never succeed. But now, because of the bigger data, much bigger data with more accurate. With more accuracy, and also this protein language model and transformer model, I think this would be the definitely next generation technology. Yeah, so this will be the final slide. So as I said, we just started the new division called medicinal frontier science. But this new approach will be very, very helpful on the basis of the new AI breakthrough. So these are always related to the our measurement. So we should be also be careful how better things happen in the different field. Yes, I think is yeah. Yes. Okay. So finally, I will show some slides. So next year, I will organize one of the mass spec conference. Is as she introduced, and now president of the Mass Spec Society of Japan. So we will host this conference. It's Asian Oceania Mass Spec Conference, located in Ishigaki Island. It's very close to Taiwan and mainland China and Korea and Japan. It's like hub location. Very nice beach. I don't know, it will be better in Thailand. But, so this is actually me. And the Ishigaki Island is very famous for scorpion. So they have their own scorpion. So I just, you see this one? It's very small, by the way. And it can attack to the human. You can feel something, but we don't have any effect. But for insect, it will kill immediately. So, and this is a peptide. So that's why we are very interested to see. And this peptide we also modified a lot. And these are also very nice target for our mass spec analysis. And this cannot be done for nanopore sequence. So that's my final message. I think mass spec-based proteomics will be still the mainstream in this field. Yeah, thank you very much. This is our current member and members 2 or 3 years ago. And thank you very much for your attention.
Not my apartment.
So now is a time for question. So if you have any question, please raise your hand. Okay.
I can send it to you and you will ask them if you want. But
you have so much data to view, so I actually want to ask the first question. You said that when you do the peptide, the 7 amino acid sequence is enough to see the uniqueness of the peptide and uniqueness of the protein. So how is it possible, even if you degrade and you have only C-terminal peptide, and usually C-terminal peptide, for example, in antibodies or we have a conservative, right, things. So how can you say that these peptides is only from this protein and this peptide only from this protein? I'm sorry, I'm not the mass spec guy, so that's my question. And another question, you said that transcriptome in every single person is different, right? And then you show us that you want to compare the healthy individual's transcriptome and COVID patient's transcriptome. How we can compare this and overlap these two groups if the transcriptome is so dynamic system?
So it's not the transcriptome, it's a peptidome. Yeah. So I think that the sequence itself is the same from the patient and also from the healthy volunteer. But because of the protease activity in patient, maybe in blood, will be different. And more peptides are like flow into the blood after infection. So that's what we estimated. And so this case, we did not use any trypsin digestion. It's just the peptide as it is. So that's why we can say this. Otherwise, it's impossible. And the C-terminal uniqueness of the protein, actually, we checked using the human proteome database to see how unique they are, especially for C-terminal sequence. I think it's unique enough. You can check. So maybe we don't need 7 amino acids because the position is already fixed. Maybe 6 amino acids will be enough.
Okay, thank you so much. Maybe can I ask one more question about the? So you said that when we use the monolith,
Silica monolithic.
silica monolith, we have sharper peak and less retention time, right? But as I imagine in my head, maybe you can explain more. As we have a monolith layer, our molecules can make more interaction when in monolith rather than we have a particle. So how is it possible that the peak is sharper and retention time is smaller?
Okay, so one thing, the peak sharp peak effect is not related to the monolith or particle. It's a common effect. If you use the very steep gradient, the very sharp gradient, the peak width becomes sharper. So it's not related to the monolith. It's more common. And for monolith, so also depends on the skeleton size, also the pore size on the surface. And the skeleton size corresponds to the particle size. So if you have the very thin skeleton, then it would be the equivalent to the smaller particle. But the current pressure, if you use a very small particle, the current pressure increase a lot. But for monoliths, you can also control the true pore. So you can use the same skeleton, but larger true pore or smaller true pore. So true pore size is highly related to the current pressure. And separation efficiency is related to the skeleton size. So now it's independent. But for particle pack, you cannot be independent. So these two factors are always related. So if you can use the smaller particle, then you can get higher current pressure. So that's the secret, I think.
Thank you.
Thank you for your nice presentation. I actually have several questions, but the way you explain it, it's easy for me as a newcomer to get the big picture. Yeah, most of my question is already answered in the next slide of your presentation. So in some point, I stopped making a question. But there is a question left. The first one is, how big the data of our proteomics that are already covered now in percentage?
The confidentness of the published data set, you mean?
Yeah.
Okay, so there are some community effort to make the data more confident. So because previously we had a lot of problem about the uncertain or false positive IDs using the very large scale experiment. So Now we have some kind of like standard rule how to make the data confident. So not for like 7 amino acids, for example, if you want to be very confident, the minimum length of the peptide
So
should be
it's
10.
like a dormitory.
This is one of the way to increase the confidence, and also the number of peptides per protein. If it's the single one, then it's
My
less
internet
confident.
is so
If you
slow.
have like more than 1 peptide per protein, with both are like more than 10 amino acids, then it's very confident. So this kind of rule
If
we already
it's not
established.
good, you can
But
find
not only
another
for
one.
MS related, we can also use another types like detention time of the LC, because also with the help of this very nice deep learning, we can very accurately predict the detention time. Then the difference between predicted and experimental one will be also the nice parameter to increase the confidence. Yeah, so this kind of like approach could be used.
Thank you for your answers. And my second question is, not a question, but it is an idea, but it's from a newcomer. It might be biased. Yeah, I see the procedure of the protein to become a fragments and then be analyzed. Maybe how if we implement a parallel process in the system so that it can prevent the delay for all samples so that they can like quickly be measured. Yeah, how about that?
Yeah, so in LC level, this is possible, like dual count system. So they already have some commercialized system as well. And within the mass spec, like ion mobility separation is one of the way also, just before tandem mass spec, to change the time to go into the tandem mass spec. So you can also separate within 100 milliseconds. So this is also really nice separation tool before this kind of like MS1, MS2 based proteomics. And also, I don't explain too much about the mode, but the DIA called data independent acquisition is always use the like wider range to collide the ion for MS. It means that so previously people want to isolate the precursor ion for MS. Then isolation should be very specific to some particular ion. Otherwise, the MS contain a lot of like non-annotated peaks, which will also decrease the confidence. But also with the help of AI, now we can treat the MS spectra from multiple precursor ion. We call chimera spectra, because contain a lot of different precursors. We can actually solve this kind of problem. So yeah, there are a lot of techniques, and yeah, but of course we need more powerful approach to get rid of the existing problem. Yes.
Thank you for nice answer.
I think we have time for the last question. Who want to ask the question? Okay.
Thank you very much for a very wonderful presentation. As he said, every question we had in mind was already answered in the next slide, and then you just answered about the AI. For me, my question is, the world of proteomics is quite complex as you presented, and is it possible to delineate this data set into a kind of network that would allow us to predict the functions of the proteins that we have analyzed, and how can AI be used for such predictions? If you are familiar, we have I think yes, you are familiar with it, the geontology and the Kite pathways. How can we play around this concept as such we could get results that would be not only meaningful at the cellular level, but also clinically?
Yeah. Like previously, we always just used like gene ontology analysis, like GOMS analysis, because this is very easy, already existing for transcriptome analysis. So we just use the same tool, but for proteomics data set. And also like metabolome analysis, for example. So metabolite map is already complete. So it's very easy to do a like network analysis based on this map. But for proteome, so there are a lot of like protein-protein interaction map there. So people use this protein-protein interaction map as a like template to do network analysis. So sometimes it works, sometimes it doesn't work. So we don't have any golden standard way to do this post-data analysis, post-acquisition of the mass spec data. Many people are doing different things, but I'm sure that this AI approach could be also very useful because experimental data set become more and more like piling up. Especially like phosphorylation, like time course phosphorylation data set, the phosphoproteome data set. This can be very useful to apply this kind of AI. Like phospho signal just go up within like 5 minutes for one particular protein, but then the downstream protein phosphorylation should occur later. So like time-wise, so this is a very typical example. Most of the cases it's very complicated and it's very difficult to analyze by our brain. So of course we need at first some kind of template data set, but once it is established, then I think it's not so difficult. Yeah, that's my estimation.
All right. Thank you very much, Professor, for a very nice talk. And thank you, students and faculty who comes today. I hope this talk spark some inspiration and be fruitful for your future research effort. So thank you very much. And if someone would like to make a connection for the further collaboration, so we have a few minutes before we move to the next activity. So you may come in front of the room and talk to the professor. Oh, okay, all right. And another thing, let's take a photo together. If you have time, please come ahead, quick. Thank you.